So we reach to the portion that is what we have seen earlier, and what are the situations, what are various principles of drug action, what are the basics of drug action, and just a minute, I have. Yes. So we have reached to the point that is what are the receptors? Receptors are the protein structures which are present on the mammalian cell or within the cell. We have seen the examples at various places where the receptors are there. Receptors are the sensing elements in the system of a chemical communication that coordinates the function of all the different cells in the body. We have seen that there are various chemical messengers which are present in the body. They are hormones, they are neurotransmitters or other mediators like autocoids, histamine, 5-hydroxytryptamine, and there are so many. So there are so many biochemicals are present in the body, and they carry various informations by means of their chemical structure, and because of that chemical structure binding or that chemical moiety binding with something which we call as a receptor, it initiates the action that we have seen as a physiology of hormone, physiology of neurotransmitters earlier. Example, acetylcholine, adrenaline, and many hormones last year you have studied. Now the same thing is there when these agents, the agents which are present inside the body, neurotransmitter hormones, if they can act, then something can be acting on these receptors, and because of that it can either increase or decrease in the action. And same principle is followed in the receptor mediation drug action. Okay, so they can act as agonist, they can act as antagonist. Okay, and on these mediators the drug will be acting. Now what happens, we have seen the definition of agonist and we have seen the definition of antagonist. So agonist, we have seen that these are the drugs which are having both capacities, that is they do have affinity and they have the intrinsic activity. Okay, so because of this variety of actions they can cause it directly. Particularly agonist is binding with the receptor, and if the receptor is on the cell membrane and that is acting as a transporter, then immediate action it will be showing, that it will be acting on a transporter or channel, and because of that it will be opening. Because of opening, it will allow the passage of certain thing. So there are such many transporters are there. Example are most easiest are example, if you have seen the acetylcholine that acts, because of that it causes activation, and because of that the voltage gated ions they open, and therefore there is a muscle contraction. So these are very fast actions. Okay, so whenever this kind of their ion channel type of there, in that case they are very fast, because immediately within certain milliseconds the actions can be there. While the second one is there, that is having a transduction mechanism. Again, if you remember, I have asked you the question earlier, that is what do you mean by second messengers. Anybody remember second messenger? Have you studied? Yes, what is second messenger? Any example? Yes, cyclic AMP is a second messenger. Very good. Very good. Okay, so cyclic AMP is a second messenger. Have you seen any third messenger? Okay, in case of the pathway of acetylcholine or noradrenaline, you will see that particularly in acetylcholine and smooth muscle, it causes activation of it causes activation of the yes, calcium. Very good. It is not extracellular; it is intracellular calcium. But Dheeraj, okay. So you have seen IP3-DAG pathway, that is diacylglycerol and inositol triphosphate. So second messengers are IP3-DAG, and they are causing opening of intracellular calcium ion, and so that the calcium from the sarcoplasmic reticulum will come to cytoplasm. So this is here the calcium becomes the third messenger. So what I mean to say here is there can be such activities where second messenger, third messenger is there. Now you can think about it logically. The first action was direct on cell membrane bound transporters. So immediate sodium ion comes in, immediately there is a change in voltage, immediately there is a change. So they are taking in milliseconds. Okay. Then this is be causing transmembrane activation. Because of transmembrane changes, there is a activation of second or third messenger. Now it will be somewhat slower than the first one. Okay. And lastly, the drug which are acting on nuclear receptors, it may take much more time, because it can take hours, or it may take days, or it may take even years to show the action. So this is how the timing is also important, how the receptors they can have a different kind of time of action. Okay, so that is how we call it as an onset of action. When action starts, we call it as an onset of action. So onset can be delayed one in certain cases. Then we have seen what is antagonist. Antagonists are drugs which are having, what is the definition? They don't have any intrinsic activity. If they don't have intrinsic activity, we have seen the example of car and driver. So if there is no intrinsic activity, this drug will not able to act themselves. But what they can do is, just like a person who doesn't know driving can go into car and will not allow the actual driver to drive the car. So definitely the activity from the car will be slowed down. Or similarly, this will happen. Yes, they have affinity and they don't have intrinsic activity. Very good. So because of that, since they do have affinity, they don't have intrinsic activity, they can just bind the receptor. But what will happen because of the binding with the receptor? They will not allow the endogenous chemicals, the endogenous hormones or neurotransmitter to bind with the receptor, and if it is not able to bind, they will not able to show the action. So this is how the antagonists, or this is how the blockers are used in therapeutics, or they are useful in pharmacology. So the drugs which are acting via receptors, they should able to act at low concentration. They always show a structure activity relationship. If small variation in structure is there, this will not open, and therefore it is very commonly called it that it is a lock and key mechanism. Just like, although locks look similar, maybe from same company, but they do have different set of keys, and one set of key will not be able to open the another lock until and unless the what they call it as a lever, until and unless these levers are matching. Similarly, until and unless some structures are matching, this will not allow, these agents will not allow any action till it is there. Then they can be antagonized by specific antagonist that we have seen. Very important thing is there that there are two components are there, therefore in each drug. That is, one component is there which is very must, which is essential for binding of the drug to the receptor. So that component is there, then and then only drug can be bound. And the second component is there which is very much important for making its active, or having the intrinsic activity. So one thing you should keep in mind, that there is a one component of any structure or any drug. That drug have a structure, and that structure have one important component that we call it as a responsible for affinity portion. So when that portion is there, when it is matching, then it will able to, just like lock and key, again I'll give you example. If you will see various kind of lock, there are certain locks which are having some slot inside. So because of that, the key must have that particular sides hole or strip in between. If it is not there, the key will not be able to be inserted. So the keyhole will have the certain structure, so that it will allow only certain type of key. So maybe lock, if they are from same company lock, then you will able to find that key can be entered inside, but it is not, it may or it may not able to open. So if it is able to open, then we call that it is having intrinsic activity. If it is not able to open, that will just go inside the lock. Until it is inside the lock, the actual key, the original key, will not able to open. And therefore many times the example you will find in book is lock and key mechanism. Okay, so that means that affinity is a portion where it should able to match the keyhole and get into it. And second thing is, the actual levers are there which will able to match with the internal mechanism, so that it will able to open the lock. So this is how the pharmacodynamics of receptor mediated drug action is there. I hope this much is clear. Any difficulties so far? Okay, fine. So next kind of the first type of receptor, they are ion channel receptors. They are called as a ligand gated ion channels, because you know now what is ligands are. Ligands are what? The agents which are having capacity to bind. This capacity doesn't mean that they are agonist or antagonist, whatsoever they may be. Okay, so these are called as a ligand gated ion channels. They are having a receptor, and they open only when the receptor is occupied by an agonist. Okay, here agonist, as I told you, in many cases they are biological compounds, they are present inside the body, and since they are present inside the body, they are the agents in body which are doing this job. So they are getting by different mechanism, like voltage gated ion channels, they are important. The drugs can affect ion channels function by interacting either with the receptor site or ligand gated channel, or with other part of channel molecule. Just like that, there are some locks where the key is something, or before key something has to be done. Okay, so the receptor before binding with the receptor, they will make some changes at some other place, but that will cause opening of the channel. Okay, so this we shall be studying particularly in antagonist when we shall classify them. But just remember that it can do, the class is benzodiazepine. Example, benzodiazepines are acting on GABA receptor, GABA type of receptor, yes. So these GABA receptors are having a role, that is GABA is one of the neurotransmitter in the brain. So this GABA, when binds with the receptor, that will cause opening of chloride channels, and because of that it will cause inhibitory type of response, that is it will cause decrease in the nerve conduction impulses. However, benzodiazepines, they bind with a different side than that of GABA. They are not actually binding to the GABA receptor, but they change the structure in such a way that it causes increase in the activity. Okay, so initially it was supposed in long back, almost 50-60 years back, it was supposed that they are acting on GABA receptor. Then later it was confirmed that yes, no, they are not acting on GABA receptor, they have their own separate receptor, they called as a benzodiazepine receptor. But these benzodiazepine receptors are able to change the GABA receptor positively, so that the GABA can act, even small concentration GABA can act more. So like this we can see such many receptors are present where we call it as allosteric site. We shall discuss it, you just keep it in mind, or while taking notes you keep, they are called as allosteric sites. So they may not be actual site, but on it this is acting. So we have seen the interaction can be direct or it can be indirect. Direct, we have seen it is directly opening ion channel, it is acting on ion channel or nearby protein, and because of that it is causing opening of ion channel. Because of opening of ion channel, there is a transport of ions, and because of transport of ions, the impulse conduction or muscle contraction taking place. This is what you have seen in physiology also. And there are other one which we have just now discussed. Example is interaction can be indirect, where it is involving a G protein. Because of G protein, it is causing, example, IP3 DAG pathway, and because of that it is opening. Okay, so this is how it can be mediated. So it can be directly mediated or indirectly mediated. Okay, then next thing is which are the agents which are acting, how they are acting. Example of these are voltage gated sodium channels are blocked by local anesthetics. So there are drugs they will block, and there are many such agents. They are acting in arrhythmias, they are used in epilepsies. In various cases we are blocking them directly of sodium channel. Because of that sodium will not enter. Because sodium is not enter, the impulse will not be generated, and because impulse is not generated, it will be slowed down. Okay, so this pain carrying impulse will be slowed down. Then dihydropyridine is a vasodilator drug which will inhibit the opening of L type of calcium channel. Because of that, if it is not open, calcium will not come in. Calcium is not coming, then it will not cause vasoconstriction, and because of that the vessel will be dilated. Because of that, blood vessel is dilated means it is causing vasodilation or decrease in the blood pressure. Then benzodiazepine I have seen, it is bind to region of the GABA receptor, just now I explained you, and because of that it will cause opening of chloride channel ions. And sulfonylureas are there which are anti-diabetics. They act on ATP sensitive potassium channels, and because of that they are causing insulin secretion. So these are the agents. So we can have variety of agents which can have openers or which are activators which causes opening. There are blockers which will cause blocking, and there are modulators which can increase or decrease in the opening of the channels. So these are such variety kind of activities we have. Then next are enzymes. Enzyme you have studied well in molecular biochemistry. You know about enzymes. There are variety of enzymes are present in the body, and they are very important part, or these enzymes are playing role in many of the activities. So there are many drugs which are targeting on enzymes. You know from biochemistry that all enzymes are proteins, and since they are proteins they have some specific structure. Since they have some specific structure, so they are not receptor actually. Let me tell you very clearly. So they do not come under receptor mediated actions, but since the enzymes are similar to that of receptor, like that means structure activity is important. Until and unless there is a specific structure, enzyme will not able to bind with that receptor. Okay, and therefore some of the books, some of the people, they put it into this kind of category. But in receptor mechanism, enzymes are that way. In many classical books you will not find that they are classified under receptor mechanism. Okay, so enzymes are separate class because they are not that way receptor mediated actions, or they are not exactly cellular mediated one. In some book they put it as a cellular one, receptor one. Anyway, our important is that is one of the method of drug action, and this method of drug action is having the role that is either activation or increase or decrease in the enzyme activity. So there are many drugs which are targeted on enzymes. Example, if you remember, again you go through physiology, you can remember that the example, if you have studied ANS, in ANS you have found that if it is adrenaline, it is metabolized by an enzyme that is known as MAO, that is monoamino oxidase. There are drugs which can block monoamino oxidase. Then you know that acetylcholine is metabolized in the synapse by the enzyme that is called as acetylcholine esterase. So wherever we want to increase the acetylcholine activity, we can give the drug which can inhibit the acetylcholine esterase. Because of inhibition of acetylcholine esterase, you can get more action of acetylcholine. Then next is there are drugs, they may act as a false substrate, although word is used as inhibitors, but what happen is they are actually acting as a false substrate. Just like we have seen the case of receptor. Similarly here, if there are the passengers, okay, in car, and suppose enzyme is a car, okay, and in that there are 4 persons are sitting. Now what will happen is, we have got a set of people who are actually need to be getting transported, which are important, which are required to be getting transported. However, if suppose, and in each car there are 10 persons, suppose, and in each car we can transport 5 persons. So what will happen is, we know that there are 10%, we need 2 cars. What will happen now, suppose there are 10 more person comes, although they are not supposed to go, but they show similar or they wear similar type of clothes or a false identity card, and they sit in the car. Okay. Now it will depend. If all 10 sit into that, then all the 10 which should have gone for the work, they will not go. Okay. That means their work is inhibited. They are not inhibited, but their work is inhibited. So this kind of things are there. We call them as a false substrate. That it was not substrate. The drug which is producing itself as a false substrate will get metabolized by enzyme, but will not allow the actual substrate to get metabolized, and since actual substrate will not be metabolized, the action will not be done. So this is how many anti-cancer drugs are working, or even we shall see that they are called as a anti-metabolites. The word is used in chemotherapy and cancer treatment and viral treatment. They are called as anti-metabolites. So these are the drugs which are anti-metabolites. The word is used as a enzyme is inhibited, but it doesn't exactly inhibit, or it doesn't stop enzyme from working. Just like we are not stopping car from working, car is going, but instead the actual person who are important, who should have gone, the other persons are sitting, and because of that the work has not been done. I hope you understood the word inhibition here. Here inhibition means the action is not blocked. I repeat, here it means that the action which was supposed to do the changes is not happening. The action is going, the car is going, but the person who should have sat into it, they are not sitting and somebody else is sitting. Okay. So this is how the enzymes are working, or false substrate are there. So this describes enzymes or enzyme inhibitors. Then there are false substrate. So abnormal metabolite is produced, and this metabolite is either having a negative role, or it is having no role. So but that has reduced the actual products. And the next is prodrug, where the drug is actually activated. So if you inhibit such kind of enzyme, the drug will not be produced. Then next in that case is drugs acting on carrier molecules like transporters. Just like ion transporter, there are some other important organic molecules are also transported. They require carrier proteins, and since they are permitting molecules, they are often too polar. We know that in absorption, that very polar drugs are actively transported, and you need some carrier mediated transported or active transport mechanism. So there are many examples of the carriers like glucose amino acid transporters are there, ion transporters are there, organic molecule transporters are there, neurotransmitter precursors like choline are there. Okay. Then neurotransmitter for noradrenaline, 5-hydroxytryptamine and glutamate, they are okay, they are taken back. So these transporters are blocked. We call them uptake inhibitors or transport inhibitors. Just like I'll finish this and then I'll answer Sava. Okay. So there are amine transporters are there. So many such drugs are there. Again, if you go back in physiology to understand the transporters, then what will happen is, you will see that particularly in case that noradrenaline, when it is released in the synapse, some of the noradrenaline is taken back into its presynaptic neuron. Why it is taken back into presynaptic neuron? Because it is required for the further synthesis or repeat synthesis. Okay. If we block this transporter, then the amount of neurotransmitter going back will be less. Initially it will increase the concentration in synapse, but eventually it will cause a decrease in the formation of neurotransmitter, and slowly it will cause decrease in that activity. So this is how the transporter blockers they are acting. Okay. So we have got several such transporter families. There are many families of transporters are there, and these drugs are blocking. Example are cocaine is there, omeprazole is blocking the we call it as a proton pump. Cardiac glycosides are blocking the transport of calcium. So there are many such tricyclic antidepressants are the drug which causes the block of uptake of neurotransmitter noradrenaline into synapse, and therefore they decrease the this level of neurotransmitter inside the synapse. So this is how they are acting. Okay. Now SABA prodrug are what? The prodrugs are the drug. Anybody knows prodrug before I answer? Let me understand. Let me know. Tell instead of writing immediately you start writing answers. Yes, Anirudh. Yes, very good. Perfect. Okay, SABA. So these are the drugs which are actually inactive in situation, and they are activated because of the metabolism in the body. Although drug I will tell you later, or you can understand later, but just like if you have studied digestive system SABA, okay, the digestive enzymes are actually inactive in nature when they come in contact with hydrochloric acids. Okay. So the pepsinogen is converted into active pepsin. So pepsinogen you can consider it as a prodrug, and when hydrochloric acid comes into contact and it metabolizes, it becomes pepsin. So that pepsin becomes a drug. Okay. Similarly, there are many drugs which are otherwise inactive, and when it is getting inside the body, when liver or any other liver enzyme or any other enzyme at any other place that metabolizes, then they are converted into drug and then they are active. Okay. This is called as a prodrug. Okay. Now it's clear, SABA. So types of receptors, there are many different type of there, we know about it, and therefore we have seen, I was telling you, now I have compiled it. We have seen if it is a ion transport type of there or transporter type of there, it will be acting in the millisecond. If you remember, I told at that time. Okay. Catecholamines, it will take some seconds. Thyroid type of hormones, they will take hours or a day. Peptides or glucocorticoids, they will take even more longer time. So we have got range of the receptors mediated action, which is from millisecond. Millisecond means, anybody? What is millisecond? Yes. What is millisecond? It is thousandth of the second. So 1 upon 1000 seconds is equal to 1 millisecond, or 10 raised to minus 3 second. So this is 10 raised to minus 3 second is 1 millisecond. So some of the drug can show action in milliseconds after binding to receptor. Yes, after binding to receptor, while some take some second, some take hour or day, some takes longer time. You understood now on what basis it is there, on what basis it is taking time. If it is ion channel, immediately ion channel open. If it is voltage gated, immediately sodium comes in, and because of there is a change in the potential, the action is immediate. Okay. But if it is the second messenger or third messenger, it will take some seconds. Okay. But if it is entering cell and it is modifying the function, it may take hours or a day. Okay. And there are certain peptides which will take much more or longer time in action. So these are how the drugs are acting or drugs are taking time in showing action. Okay. Next are the structures are there, how they are showing or how fast they are acting. Example is, we call it them as ligand gated ion channels. We call them as ionotropic receptors. So they are membrane proteins which are similar structure to ion channels, and they ligand binding receptor site present in the extracellular domain. So when it acts on the receptors, it causes opening of the channel. Because of that, it causes either hyperpolarization or depolarization, and because of that, it is there. The time taken is milliseconds. Example, nicotine acetylcholine receptors. Okay. So they are having similar function that of nicotinic acetylcholine receptors. It will bind. This is the receptor structure, and these are the alpha, beta and delta types of proteins are there. Okay, these subunits they are called, these protein subunits are there, and they are forming the structure like this. So when acetylcholine binds with that, it will cause opening of the receptor, that will open, and the pore size that will be bigger one, and because of this pore size, you can see pore 0.7 nanometer diameter. So that will open, and so that it can cause easily passage of the ions which were otherwise not transported. So this is how acetylcholine receptor is there, and this is how it causes immediate action. Okay. Then the next are G protein couple receptor. We shall see still more details about it, and you have seen about them. There are having the second messenger mechanisms. So they will either act through ion channels, or they act through protein phosphorylation or through calcium release. Okay. So these are variety of action it can cause. Example is muscarinic receptor of acetylcholine. Then next one is next one type of receptor are kinase linked receptor. These are the large and heterogeneous group of membrane receptors. They are again extracellular type of ligand binding domain are present on that, and it is enzymatic nature in nature that causes the activation of protein kinase or guanyl cyclase. And this receptor example is for insulin or cytokine type of receptor. So when it binds, it causes protein phosphorylation, it causes gene transcription, and because of that protein synthesis, and thereafter cellular effect. So you can understand that therefore it is taking, we have seen protein type of receptor or peptide type of receptor. Insulin is a peptide. So peptide type of receptor may take days or years to show the action, because when it will cause gene transcription and protein synthesis and then cellular effect, that much time it will be taking. Okay. So this is how various receptors are there. Lastly, nuclear receptors. These are the receptors that regulate the gene transcription. You have seen about glucocorticoid or steroid hormone receptors. So they will get inside the nucleus. In nucleus, they will bind with the DNAs, and from that it will cause gene transcription, and accordingly particular sequence of gene will be processed, and that will cause protein synthesis. It will cause cellular effect. So this is how they are there. There are a variety of such steroid receptors are there. then thyroid hormone is acting through it this way, then other like retinic acid, vitamin D, they all are acting, because vitamin D also is a steroid. So all, then sex hormones, everything they are acting through this mechanism. Okay. So this is how the drugs are acting. Okay. So here we have seen what kind of actions drugs are showing. Okay. And it started long back in 1909 by Paul Eldridge, and as I told you, he was the person to tell about these two things. It should have a affinity and it should have a some unit which will show activity. Okay. So these are how various receptors are there, and important now in this is how the drug receptor interactions will be taking place. We shall go for these drug interaction activities in the next few classes. They are called as a theories of drug action. Okay. They are called as a rate theory or occupation theories. Okay. So how these are there? Actually, they are important only for few kind of receptor, but yes, they play very important role. Okay. And some of the terms we shall try to understand from the again basics of affinity and intrinsic activity. We have seen this figure already, what agonist is and what antagonist is there. What do you mean by affinity and what is intrinsic activity? This we shall be using further for having few more terms to understand. Okay. So in that we shall understand what do you mean by potency, what is mean by efficacy. Okay. And then we shall able to go for further more details about what are the actions of the drug, or what are the various theories that makes the action of drug. This much is understood. Is there any difficulty in understanding concepts? Even we will try to go for understanding receptor concept in more details. Okay. So far you please concentrate on this much knowledge you have got, and as I ask you, please go through it, please go through, please read it, because the depth is too much. When I am explaining, if you understand the basic point and the concept wise, then reading will be easy. Anybody have started reading? Yes, Anirudh, it happens. No, therefore, okay. What is first pass effect? What is first pass effect, Anirudh? Okay, good question. Let me understand. Okay. Yes. Just a minute, hold. Let me answer a call. I'll join you. हेलो, फ़ोन इसलिए किए थे कि तू कॉलेज अगर पहुंचा होगा ना, तो एक बार ये देख लेना। एग्ज़ाम रूम में मैंने खुर्गे जी को फ़ोन कर दिया था कि साफ़ सफाई उस रूम की देख लेना करके। खुर्गे बोल रहा था मैं फ़ोन कर दूंगा उसको करके। हाँ, तो वो किसको, दोनों को कर दिया ना? बहुत बहुत धन्यवाद भैया, चलता है, इसीलिए फ़ोन किया। थोड़ी नहीं, ज़्यादा लगते थे। हाँ, वो मालूम है मेरे को, ऊपर का सब देखा फिर। हाँ, ऊपर का सब अच्छा दिखा उनको, फिर उन्होंने फिर मार दिया, ऐसा है साफ सफाई नहीं। वैसे 2 बार साफ करके गया था वो, फिर मेरे को क्या बोले वाला, मैं तूने उसको यादव को क्यों छोड़ दिया? मैंने कहा मेरे को पूछ के नहीं गया, वो तो बोल रहा तेरा नाम बोल रहे, तेरे को पूछ के गया। एक चक्कर लगाऊंगा मैं थोड़ी देर के बाद, है ना। और अगर भीड़ हो गई बाई चांस तो मेरे को बोल देना, वैसे तो फ़ोन कर देना। हाँ, मेरे को भी नहीं लग रहा है, मैं थोड़ा दूसरा इधर उधर के काम करके फिर आऊँगा मैं, है ना। ओके, चल, धन्यवाद भाई।